We discovered, fortuitously, that we could quite easily distinguish an embryonic stem cell that had come from a naturally fertilized embryo or a somatic cell nuclear transfer embryo from those that have been generated by parthenogenesis without the contribution of fertilization. And that's critical because in attempting to make cell lines by nuclear transfer, one can actually, by mistake, isolate parthenogenetic lines. In a typical somatic cell, you'll have a copy of both the mother's gene and the father's gene. Whereas in a parthenote, you'll have 2 copies of the mother's and 0 copies of the father. The predominant misconception in the scientific community was that a cell line that was derived by parthenogenesis should be what's called homozygous. That is, it should have taken the copy of the mother's DNA and duplicated it entirely so that all the genes all along were identical. And yet we know from our work that that's actually a misconception. That in fact, the way most parthenogenesis is done leaves the cell line with actually a majority of mixed up genes from the mother and the father, what we call heterozygosity. When you're trying to determine whether 2 biologic samples are related, you look for these so-called single nucleotide polymorphisms, or these subtle genetic changes that distinguish the mother's genes and the father's genes. Well, what we've done in the past is we've looked at a relatively small set of those markers, maybe 3 or 4. And when you're only looking at 3 or 4 markers, you don't have as much resolution. You don't have the ability to say with great confidence that 2 samples are related or are unrelated. So what has come through the Human Genome Project is now availability of thousands and thousands of these markers. And so you can actually analyze across the entire genome, in our case, 50,000 different markers, which gives you an enormous degree of genetic resolution.